Doctor! Doctor! My human baby is so lethargic and pale, and its abdomen is distended. What's wrong with it?
That's an easy diagnosis. Your child has beta thalassemia.
What's that?
Well,
Beta thalassemia can be categorized as a group of inherited blood disorders that is estimated to affect 1 in 100,000 people globally, with significant populations in the Mediterranean, Middle East, Europe, Central Asia, and countries along the northern coast of Africa. The disease results in the production of malformed hemoglobin in the red blood cells, which can cause anemia. Hemoglobin is an iron-containing metalloprotein responsible for the transport of oxygen from our lungs to the rest of the body. It consists of 4 individual protein subunits, 2 alpha globins, and 2 alternate globin chains. These alternate chains exist as gamma globin in infants of 0 to 6 months, which converts to beta in adults through an unknown conversion process. The biochemistry of hemoglobin focuses around heme groups, which are nitrogen-containing structures covalently bound to iron 2 ions, which regulate the transport of oxygen and carbon dioxide. The human hemoglobin alpha HbA gene is located on chromosome 16, while the human hemoglobin beta HbB locus is located on chromosome 11. Contained within the beta locus are the genes responsible for the fetal gamma and adult beta globin chains, which are tightly transcriptionally regulated to one another. The disease is inherited in an autosomal recessive manner, but often presents as autosomal dominant. This is because the disease can be caused by smaller point mutations denoted as beta plus, which can cause misforming and misconformation, decreasing binding affinity of oxygen to hemoglobin, or deletion mutations denoted as beta naught that create non-functional protein chains unable to be incorporated into the hemoglobin tetramer, abolishing binding altogether. Hereditary combinations of the mutations can result in different severities of the disease, which are classified as thalassemia major, thalassemia minor, and thalassemia intermedia, diagnosis of which is based on the severity of presented symptoms. Most common symptoms include iron deficiency, jaundice, pallor, splenic and liver enlargement, as well as muscular atrophy. In thalassemia minor, HbB chains have small point mutations that decrease the binding affinity of oxygen. However, these individuals usually present as clinically asymptomatic. In thalassemia intermedia, HbB chains have almost any combination of small point and larger deletion mutations that present clinical symptoms of intermediate severity. Beta thalassemia major, also known as Cooley's anemia, is characterized by the complete loss of beta oxygen binding, causing crippling symptoms. There are 2 main treatments for beta thalassemia, those being bone marrow transplants and red blood cell transfusion. Bone marrow transplants force the body to produce normally functioning HbB from donor marrow. However, it is an extremely invasive procedure and donor compatibility is rare, even within familial bloodlines. Red blood cell transfusion is a preferred method to transplantation, as it is a less invasive and simpler way to introduce functional HbB to the bloodstream. However, under these circumstances, the body does not produce the protein itself, and red blood cells only have a 115-day viability period, after which additional transfusions are necessary. In conjunction with the time-intensive nature of the procedure, multiple complications also arise. Issues such as post-transfusional iron overload occur due to the gradual buildup of iron in the epithelial tissue and macrophages within the body. Iron buildup can lead to issues with general growth, delayed sexual maturation, as well as impaired endocrine function, which is why the treatment has been implicated in causing diabetes and both hyper as well as hypothyroidism. As a result, researchers have begun taking advantage of the gamma to beta conversion pathway by using fetal hemoglobin as a new disease treatment. It has been proven that people with beta thalassemia naturally compensate for the lack of functional beta globin by overexpressing fetal gamma hemoglobin, the presence of which has been seen to ameliorate symptoms of the disease. In more recent studies, microRNA expression has been used to target specific transcription factors implicated in the gamma to beta conversion pathway. Expression of the miRNAs leads to the suppression of the transcription factors in the human hemoglobin beta pathway, thus upregulating gamma expression. Additionally, genome editing of the HbB gene in induced pluripotent stem cells has been proven to produce functional HbB in vivo in humans. However, it is also an extremely costly and time-intensive process. Researchers are now investigating the mechanism behind the gamma to beta conversion to find a long-term treatment for the disease. By understanding this pathway, it may become possible to directly induce human hemoglobin gamma production, further ameliorating the disease. Potential steps in conjunction with this idea may be to investigate the effects of persistent gamma globin presence to also better understand the purpose of the gamma to beta conversion. In conclusion, beta thalassemia is a genetic disease that can be caused by any number of mutations in the human hemoglobin beta gene, which leads to decreased oxygen binding in red blood cells. Currently available treatments, although limited, have drastically improved quality of life for people living with the disease, and with further research, it is hoped that people will one day be able to understand the disease in full.